And today we'll continue to speak about the tissues of internal environment. Look at the classification again at the tissues of internal environment. This group of tissues is subdivided into the blood and lymph. About the blood and lymph, we told previous lecture. Then the next group is the group of proper connective tissues, tissues with special properties, embryonic tissues, and also skeletal tissues. So today we are speaking about the proper connective tissues, about the tissues with special properties, and embryonic connective tissues. Look at the main characteristics of tissues of internal environment, common distinctive properties, and the first characteristic, all tissues develop from mesenchyme. About the mesenchyme we'll speak this lecture. It is the sort of embryonic connective tissue. Cells of mesenchyme form from the embryo during the period of embryogenesis. These cells migrate throughout the body and form the different types of tissues of internal environment. So the first characteristic, all tissues developed from mesenchyme. Next characteristic, cells lie separate from one another. There is a large distance between the neighboring cells of this kind of tissue. And the extracellular matrix is prevalent, is prominent. The volume of extracellular substance is more bigger than the number of cells in all tissues of internal environment. So today we'll start to speak about the proper connective tissues and look at the main functions of proper connective tissues. The first function is to provide the structural and metabolic support for other tissues and organs throughout the bodies. Second characteristic, proper connective tissues mediate the exchange of nutrients, metabolites, and waste products between tissues and blood circulatory system. Next function is the defending function and protection of our organism. And the last functions is the depot and storage of different substances, such as some kinds of connective tissues, storage of fats. Storage of fats, storage of water, storage of energy. So these are the main functions of connective tissues. Look at one more at the classification of proper connective tissues. Proper connective tissues are classified into the two main groups. The first group is named loose fibrous connective tissue. Second main group is called dense fibrous connective tissue. Dense fibrous connective tissues are subdivided into the dense regular and dense irregular connective tissues. Look at this slide, at the first picture, at the first illustration. We can see a loose fibrous connective tissue. Loose fibrous connective tissue is characterized by the many types of cells. There are 9 or maybe 10 types of cells we can meet in loose fibrous connective tissue. And also in the extracellular matrix, the ground component is prevalent than the fibrous component. Ground substance is more bigger than the fibrous component. At the second and third illustrations, we can see dense fibrous connective tissue. In dense fibrous connective tissue is represented mostly by the fibers in the extracellular matrix. There are less types of cells we can meet in the dense fibrous connective tissue, and in extracellular matrix, fibers are bigger. The volume of fibers is bigger than the volume of ground substance. In the middle picture, we can see dense irregular connective tissue in which fibers are situated disorderly, forming the structure like a net. In dense regular connective tissue, fibers are situated in correct order, usually parallel to each other. So we'll start with the loose fibrous connective tissues and look at the main characteristics in structure. Abundance of cells, jelly-like extracellular matrix, and ground substance is in the extracellular matrix is prevalent than the number of fibers. So look at this illustration. Loose irregular connective tissue are spreaded, directed in our organism. Usually all blood vessels, all nerves are surrounded by loose irregular connective tissue. Also, loose irregular connective tissue we can meet in the wall of inner organs, for example, some layers of mucosa, submucosa are formed by loose irregular connective tissues. Also, papillary layer of skin, dermis, is formed by loose irregular connective tissue. So this type of tissue is widespread in our organism. This type of tissue is characterized by presence of large types of cells, and look at the cellular populations of loose irregular connective tissue, and all types of cells are subdivided into the two main groups. The first group is called fixed or resident cells. Resident cells are constantly presented in the tissues. These are long-lived populations of cells, and this group includes the following populations: fibroblasts, different fibroblasts, adipose cells, or another name, fat cells, pericytes, mast cells, and macrophages. This is the first main groups. Second main group of cells of loose fibrous connective tissue are the transient cells. Transient cells migrate to the loose connective tissues from the bloodstream, sometimes if it is necessary, and perform their functions into the connective tissues. So these cells are usually short-lived and represented by different types of mostly leukocytes. Look at this. Plasma cells. Plasma cells are the types of B lymphocytes. Another types of lymphocytes also can migrate from the bloodstream to the loose fibrous connective tissues. Such leukocytes as neutrophils, eosinophils, basophils, monocytes are also present into the loose connective tissue and migrate from the bloodstream. Monocytes migrating from the blood to the connective tissues transform into the macrophages, and macrophages perform their functions as cells of immune system. Some macrophages inside the loose fibrous tissue are fixed, but where others are transient. So there are many types of cells we can meet, and now we'll start to speak about each type of cells. The predominant cell type of predominant cell types of loose fibrous connective tissues are the fibroblasts. Fibroblasts perform a very important function because these cells produce all substances into the extracellular matrix. Fibroblasts produce both components of extracellular matrix, different types of fibers, and also different chemical substances of ground substance. So fibroblasts synthesize collagenous, elastic, and reticular fibers, which are presented into the extracellular matrix, and also synthesize the macromolecules of ground substance. According to their function, look at the morphology of fibroblast. So fibroblast is characterized by many processes. Look at this, irregular in shape because have many processes. These processes help to synthesize the fibers of extracellular matrix. Also, these cells are characterized by the presence of euchromatin in the nucleus. I repeat that, as you know, euchromatin is the active type of chromatin. It is crumbly chromatin, and all genes are opened to the transcription. Transcription takes place, that is why translation takes place, and protein synthesis increases. So euchromatin is prevalent type of chromatin in the nucleus of fibroblasts. Also, cytoplasm. Cytoplasm is characterized by well-developed rough endoplasmic reticulum and Golgi complex. These organelles are necessary for the protein synthesis and release these proteins. Fibroblasts release the proteins to the extracellular matrix. So these are the ultrastructural characteristics of fibroblasts, but this group of cells, group of fibroblasts, include many types of cells. So different of fibroblasts are represented by young fibroblasts. These are non-differentiating cells. Then mature fibroblasts. Mature produce the chemical substances of extracellular matrix. Fibroblasts have ability to division, and when mature fibroblasts become older, then transform into the older cells, fibrocytes. Fibrocytes have no ability to division and also have no ability to produce the extracellular matrix. Also, fibroblastic differon includes another types of fibroblasts, such as, look at this, myofibroblasts and also fibroclasts. What are the cells? Myofibroblasts are the specific cells which have some similar similar characteristics with fibroblasts and some similar characteristics with smooth myocytes. Myofibroblasts have ability to produce the proteins of extracellular matrix and also have ability to contraction because cytoplasm of myofibroblasts houses the contractile filaments. Myofibroblasts take part into the reparative processes, and help to form the connective tissue scar. And the last kind of fibroblasts are named fibroclasts. Fibroclasts, also kind of fibroblasts, have ability to phagocytosis of different alien substances, performing the defending function, and also characterized by the presence of many lysosomes in their cytoplasm. So the main function of fibroclasts is the destruction of extracellular matrix in opposite to fibroblasts. So this is the main predominant cell type of connective tissue proper, fibroblasts. So next type of cells in the connective tissue are named macrophages, or another name, histiocytes. Macrophages, look at this, or histiocytes. The size from 10 to 30 micrometers, irregular in shape, look at this. Their surface of these cells is characterized by many pseudopodia, these projections. Pseudopodia surround some alien substances and phagocyte, destroy, digest these alien substances. So the main function is immunological defense of our organism by the phagocytosis of different alien substances, take part in the immune response, take part in immune defense of our organism. So these cells protect our organism by the phagocytosis and also produce the different and synthesize the different lysosomal enzymes. Cytoplasm of macrophage is characterized by many lysosomes. The enzymes of lysosomes are released outside the macrophage and regulate the functional activity of another cells of connective tissues and regulate the constancy of extracellular matrix, control the condition of extracellular matrix. Macrophages or histiocytes are formed from blood monocytes. When monocytes leave the bloodstream, leave the blood vessels and migrate to the connective tissues, they transform into the macrophages. Main function is the protection. Look at this. Main function, take part in immune reaction and produce many biological active substances. Next type of cells of loose connective tissue. Students, everything is okay? All slides are visible?
Yes, Doctor.
Sound good?
Yes.
So now the third type of cell in loose fibrous tissue are named mast cells, or another name, labracytes. The size from 20 to 30 micrometers, and these cells are characterized by the presence of many granules in the cytoplasm. We can see many granules of the cytoplasm because the main function of mast cells are the production of some biological active substances. Look at this. These biological active substances are named heparin, histamine, serotonin, and all these substances are situated inside the granules. Look at these dark purple granules in the cytoplasm. Histamine, serotonin, heparin, dopamine, and other substances regulate the permeability of blood vessels wall, permeability of wall of the blood capillaries, and also regulate the homeostasis of loose fibrous connective tissue. Homeostasis, look at this table, is a constancy of inner environment of loose fibrous connective tissue. Look at this. Granules of mast cells, or another name, labracytes, are stained metachromatically. What does it mean? The substances inside the granules have ability to change the color of the dyes. For example, we use black dyes, but granules are stained in purple color. Or we use purple dye, but granules are stained in dark brown color. So this ability are named metachromasia, ability to change the staining of the dyes. So now, next types of cells in the loose fibrous connective tissue is named plasma cells. Look at this, or plasmacytes. Plasmacytes are the effector subpopulation, one of subpopulations of B lymphocytes. B lymphocytes, as you know, previous lecture, are the kind of lymphocytes, and plasmacytes differentiate from the B lymphocytes. And the main function is take part into the immune reaction, take part in the humoral immune response. I repeat that humoral immune response is the protection of our organism against the viruses, bacteria, toxins, and other alien substances via the production of antibodies or immunoglobulins. So the main function of plasmacytes, plasma cells, are the production of immunoglobulins or antibodies. According to the gamma globulins, mostly antibodies are named gamma globulins. According to their function, according to the chemical composition, all antibodies are the proteins, complex of proteins with carbohydrates, named glycoproteins. So, the main function of plasma cells is the production of glycoproteins, and according to their function, we can see the main features, the main characteristics of plasma cells. Nucleus, very interesting in plasma cells. Look at this. The euchromatin, active type of chromatin, crumbly chromatin, is a prevalent type of chromatin in the nucleus. And also in the nucleus, we can see some portions, some parts of heterochromatin. Nucleus is called arrows in the wheels, portions of euchromatin and near portions of heterochromatin. Again, euchromatin, heterochromatin, euchromatin and heterochromatin. Looks like the arrows in the wheel. Cytoplasm of plasma cells is characterized by a well-developed rough endoplasmic reticulum. So, hyaloplasm is filled by the cisterns of a rough endoplasmic reticulum for the protein synthesis, for the synthesis of immunoglobulins. And look at this, Golgi complex is also well developed. Golgi complex situated in plasma cells near the nucleus and looks like a light spot. In the micro preparations in the slide near the nucleus, we can see the light spot, arrow indicates it is a place of localization of Golgi complex into the plasma cells. And look at this illustration, also plasmacyte, dark cytoplasm because of the presence of large volume of rough endoplasmic reticulum, and the white light spot near the nucleus, it is a place of Golgi complex. And also there are many mitochondria in the cytoplasm because this processes of protein synthesis requires the high level of energy. Next type of cell in the loose fibrous connective tissues are named adipose cells, or another name fat cells. And fat cells are very interesting cells. Very interesting cells because cytoplasm of fat cell is filled by the droplet of lipids. So all volume of cytoplasm is filled by lipid droplet. Nucleus situates on the periphery of the adipose cells, and also organelles, all organelles are also situated on the periphery of adipose cells. The main function of the cell is a storage and depot of fat, storage and depot of water, also depot of vitamins, which are diluted into the fats. A, D, E, K, all these vitamins are stored into the cytoplasm of adipose cells. When we prepare the micro-preparation, when we prepare the slides and use the spirits, usually this lipid droplet are diluted in the spirits. So when we prepare the slides, we cannot find the lipid droplet, we cannot find the droplet of fat in the cytoplasm, and cells look empty. We can see only cell membrane, only plasma lemma, Sometimes we can see peripherally located nuclei, but lipid droplets are usually invisible in the cytoplasm. Look at this. Also, if you know, lipids, fats requires the specific method of staining. The dyes hematoxylin and eosin do not stain the lipids. Usually for this indication of lipids, we use, for example, osmi, oxygen, osmi acid, or Sudan, specific dyes for the detection of lipids. in your slides, stained by hematoxylin and eosin, lipids and fatty droplets are not visible in the cytoplasm, and we can see empty cells. Adipose cells are subdivided into the two main types. Look at this. If adipose cell houses only one lipid droplet, this type of cell is named unilocular fat cell. If the fat cells houses many small lipid droplets, this type of fat cell is named multilocular fat cells. Different types of adipose tissues, according to the types of fat cells, we can differentiate, but about this we'll speak later after the break and on the second part of our lesson. So next types of cells we can see in the wall of blood vessels. For example, cells which named pericytes, look at this. Pericytes are the cells with processes and are situated in the wall of blood capillary. Look at this slide. Cells with processes, pericytes, and are situated inside the blood basement membrane. So pericytes are embedded into the basement membrane of blood capillary and form the wall of blood capillary. The processes of pericytes houses the contractile filaments, and the contraction of these filaments regulate the diameter of blood capillary, decrease or increase. Also, pericytes takes part into the reconstruction of capillary wall and reparation of the connective tissue. Inside the blood capillary, we can see the endothelial cell. Endothelial cell is a very interesting cell lining the lumen of blood capillary, very thin cytoplasm, have the very thin cytoplasm and poorly developed all common organelles. Endotheliocyte usually necessary for the transport of gases through the capillary wall, transport of different nutrients, different substances through the capillary wall, and usually endotheliocytes make the connection with the pericyte nexus, adhesion junctions, connect with pericyte by the nexuses, by the adhesion junctions, and pericytes influence on endothelial cells, influences on proliferation, migration, differentiation of endothelial cells. So these cells connect with each other and influence one to another. Also cells of loose fibrous connective tissues, but these cells we can meet in the wall of blood vessels. And one more, the last types of cells in the loose fibrous connective tissues are the pigment cells, or melanocytes, another name. Pigment cells are star-shaped, as you know, as you see, with many short, wide branched processes, and cytoplasm of these cells houses many pigment granules with pigment melanin. That is why these cells also called melanocytes. The main function of these cells is the protection of our organism from ultraviolet radiation. under the influence of ultraviolet radiation, our skin changes the color from dark to pale, more dark or more pale, because under the influence of ultraviolet, the pigment cells produce their melanin granules. So we told about the fixed or resident cells of loose fibrous connective tissue, and also different types of leukocytes. As you know, consists of two main components. The first main component is a ground substance, and the second main component is a fiber. Look at this. There are three main types of fibers we can see in the loose fibrous connective tissue. The first type is called collagen fiber. Second type is named elastic fibers, and third type is called reticular fiber. Reticular fiber is a sort of collagen fibers, one kind of collagen fibers, but sometimes we differ the third type, reticular fibers. Now we are speaking about the collagen fibers. Look at this illustration. Collagen fibers are thick, solid, and stronger. Thick, solid, and stronger, and characterized by the presence of protein collagen. Look at this. There are more than 25 different types of collagen according to their characteristics of their polypeptide chains. So 25 different types of collagen molecules, but in fibrous connective tissue, we usually can meet first type of collagen in the collagen fibers of the skin, tendons, and also third type of collagen we can meet in the collagen fibers, and fibers which made by third type of collagen are named reticular fibers. Also, reticular fibers we can see in the type 3 collagen we can see in the skin dermis, in the wall of blood vessels, and in the loose connective tissue. So collagen fibers thicker than the other types of fibers, non-branched but stronger, and characterized by the cross striation. Look at this. We can see dark and light strips. Cross striation is a main characteristic of collagen fibers. Protein collagen is the main component of collagen fibers. Another component of collagen fibers are the carbohydrate component. Glucose aminoglycans are also present in the structure of collagen fibers. There are some levels of organization of collagen fibers. Look at this, and the letter A, the first level of organization, is named polypeptide level. Collagen molecule, which is a molecular level, collagen molecule is composed of 3 polypeptides. 3 polypeptides are twisted to form the collagen molecules. So there's the first molecular layer, the molecular level of organization. Second level of organization is a microfibril level, collagen microfibril. Some molecules, collagen molecules, are connected by the cross links. Connected by the cross links to form the collagen microfibril. Third level of organization is named collagen fibrils. Some collagen microfibrils connect with each other by the glycoproteins. These bridges of glycoproteins connect the microfibril into the collagen fibers. And the last level of organization is the level of collagen fibers, when some collagen fibrils form the larger structure, collagen fiber, connect with each other also by the glycoproteins. So collagen fibers, usually we can meet type 1 collagen fibers in the loose fibrous connective tissue, thick, strong, and solid. One more kind of collagen fibers named reticular fibers. Reticular fibers are made by third type of collagen. Reticular fibers are situated in the reticular connective tissue. We'll speak about the reticular connective tissue later, to form the stroma of hematopoietic organs. Stroma performs the supporting role and forms the environment for the developing formed elements of the blood in the hematopoietic organs. So reticular fibers we can meet in the reticular connective tissue in the structure of hematopoietic organs. For the detection of reticular fibers, we use a specific method of staining, impregnation by the silver nitrate, and reticular fibers, that is why, are named ergophilic fibers, because they are indicated by the silver salts. And the last type of fibers, in the loose fibrous connective tissue are named elastic fibers. Elastic fibers are have ability to stretching, but after the stretching, they may return into their own size and shape and length. The main proteins in the structure of elastic fibers are elastin molecules. Look at this, elastin, protein elastin. Molecules of elastin looks like a worms in non-straighting condition, and elastin molecules connect with each other by the protein desmosine. So two main types of proteins. When the elastic fibers stretch, the elastin molecules becomes longer, elongated, change their shape, deformates, the elastic fibers elongate, and then return to your own shape and length. Some elastic fibers are non-mature, and these types of elastic fibers are named oxytolin and elaunin elastic fibers, non-mature elastic fibers. Then quantity of elastin is less in this type of elastic fibers than in mature elastic fibers. In mature elastic fibers, elastin is situated in the center and is occupied the center of elastic fibers. In elaunin, less quantity of elastin, white color. In oxytolin fibers, non-mature elastic fibers, the smallest quantity of elastin. So look at these three main types of fibers we can see in the loose fibrous connective tissue, but a ground substance, second component of extracellular matrix, is a prevalent component in loose connective tissue. Ground substance house the compound chemical substances, proteins, together with the carbohydrates. And these substances are named protoglycans. These are proteins, complex of proteins with glucose aminoglycans. And the second part, the second chemical substance, glycoproteins. Glycoproteins when the quantity, the volume of carbohydrates is more bigger than the volume of protein component. Protoglycans and glycoproteins are the main chemical substances into the ground substance. Look at this at first. Glycoproteins are also called adhesive proteins, such as fibronectin, such as laminin, and another types. Laminin, fibronectin are the main glycoproteins, and these glycoproteins necessary for the connection of cell plasma lemma with extracellular matrix. Fibronectin, for example, connect the cell plasma lemma with the components of extracellular matrix. Also, these proteins, glycoproteins, are necessary for the connection between different components of extracellular matrix. For example, between collagen fiber and some part of ground substance. Connection between the components of connective tissue, that is why named adhesive proteins, glycoproteins. Look at the structure of protoglycans. Look at this structure of protoglycans. The main function of protoglycans are the storage of water. These molecules of protoglycans connect the water molecules and storage the water molecules inside the extracellular matrix of connective tissue. So look at the structure of protoglycans. At the base of protoglycans, we can see the hyaluronan molecule. Look at this hyaluronan molecule from the base of protoglycan molecule, protoglycans. Small monomers of protoglycans, are attached to hyaluron molecules by the proteins. At the base, linker proteins attach the monomers of protoglycans to the hyaluronan molecules. And the monomers of protoglycan at the base, we can see the core protein, and core protein is crossed by the keratan sulfate and chondroitin sulfate, glucose aminoglycans, non-sulfatide glucose glucose aminoglycans. So one more, this structure is named polymers of protoglycans. Polymers. These polymers aggregates and attach the large volume of water and storage the water inside the extracellular matrix. At the base of this aggregate, there is hyaluronan molecule. Linker protein attach the core protein to the hyaluronan molecule. Core protein is crossed by sulfatate glucose aminoglycans, keratan sulfate and chondroitin sulfate, and each molecule of sulfatate glucose aminoglycans attach many molecules of water. That is why extracellular matrix of loose connective tissue fibers houses the large volume of water because of the presence of aggregates of protoglycans. So we told we told about the loose fibrous connective tissue, where the volume of extracellular matrix is more bigger than the volume of cells, and the amorphous component of extracellular matrix, a more ground substance of extracellular matrix, prevalent than the fibers. Now we are speaking about the dense connective tissue. In dense connective tissue, volume of fibers prevalent than the volume of ground substance. Dense connective tissue houses less cell types than loose connective tissue. Mostly we can see here the nuclei of fibroblasts and fibrocytes, the prevalent different of cells in dense connective tissue. Another difference are poorly developed. Sometimes we can meet another difference, but mostly different of fibrocytes. I repeat that in extracellular matrix, fibers, the volume of fibers bigger than the volume of ground substance, and there are two main types of dense connective tissue according to the direction of the fibers. If the fibers are situated into the correct order, parallel to each other, this type of dense connective tissue is named regular connective tissue. If the fibers are situated disorderly, like a net, this type randomly, this type of dense connective tissue is named irregular connective tissue. So a regular connective tissue we can see, for example, in the tendons, in the ligaments, and in the epineuroses. Dense irregular connective tissue are widespread in our organism, and we can meet, for example, in the dermis of the skin, the dense irregular connective tissue. Please compare loose connective tissue and dense connective tissue. Here, the upper part, loose connective tissue, characterized by many cell types, and we can see many nuclei of these cell types. Here, less nuclei because mostly fibroblasts and fibrocytes are present. And looser is characterized by the presence of large volume of ground substance. Here, the large volume of fibers we can see. Compare loose and dense connective tissues because you should recognize different types of connective tissue on your presentation on the practice classes. One more, this is the finger skin. Finger skin, upper part of the skin is named epidermis of the skin, and epidermis houses the squamous stratified keratinized epithelium. We told about the epithelium tissue. It's the first lecture. But second part of the skin, look at this. This part is named dermis, and dermis of the skin is represented by two layers. These projections of dermis are named papilla. That is why first layer of dermis is called papillary layer of dermis. Inside the papilla, we can see papillary layer of dermis, which is represented by loose fibrous connective tissue. Loose fibrous connective tissue with large number of cell types and large volume of ground substance in extracellular matrix. Below the papillary layer, there is a second layer of dermis, arrow indicates, reticular layer of dermis, bright pink color. reticular layer is represented by dense, irregular connective tissue. Here we can see the large volume of collagen fibers, which are situated disorderly like a net. Collagen fibers are oxyphilic structures stained by eosine in bright pink color. So these are the reticular layer, dense, irregular connective tissue, a large volume of collagen fibers, which are situated disorderly like a net. That is why a reticular layer of dermis. This is an example of dense, irregular, and loose fibrous connective tissue. Example of dense, regular connective tissue is the tendon. Tendon, look at this, longitudinal and cross-section of the tendon consists of the bundles of collagen fibers. Collagen fibers are situated parallel to each other and form the bundles of collagen fibers. So in the tendon, we distinguish bundles of the first order. Look at this. Bundles of collagen fibers of the first order. These are the slim bundles of collagen fibers, which are separated one from another in the tendon by the cells, fibrocytes. So between the nuclei of fibrocytes, we can see bundle of the first order. Some bundles of the fibrocytes separate the bundles of collagen fibers of the first order. Fibrocytes in the tendons are also called tendocytes or tendinocytes. Tendinocytes. Some bundles of the first order together form the bundle of the second order. Look at this. Now arrow indicates the bundles of the second order. Bundles of the second order larger than the bundles of the first order and separated from one another to the slim septa of connective tissue, slim layer of connective tissue, loose fibrous connective tissue. This slim layer of loose fibrous connective tissue is named endotendinium. So endotendinium separate the bundles of the second order. Some bundles of the second order form the bundles of the third order. Look at these large structures. Bundles of the third orders are also separated one from another by the layers of loose fibrous connective tissue, named peritendinium. And all tendon is surrounded by epitendinium. It is a dense, irregular connective tissue. So tendon houses three types of connective tissues. First type, dense, regular connective tissue. Second type, loose fibrous connective tissue, separating the bundles of the second and third orders. And houses the blood vessels for the nutrition of the tendon. And the third type, dense, irregular connective tissue, forms the epitendinium, which covers the tendon outside. This is the longitudinal section of the tendon. It is the longitudinal section of the tendon. And on your presentation, you also see the cross section of the tendon, and you should answer the structure of the tendon on colloquiums and on your practice classes. One more longitudinal section of the tendon. Epitendinium covers the tendon outside. It is a tunic of the tendon made by dense, irregular connective tissue. Bundles of the first are separated from one another by the nuclei of fibrocytes. Bundles of the second order are separated by the slim, loose connective tissue named endotendinium, and bundles of the third order are separated one from another by peritendinium. Three types of connective tissue we can meet in the structure of the tendon. So look at this classification. One more. We talked about the proper connective tissue, which is subdivided into the loose and dense. We talked about the dense, regular, these types of connective tissues. Now we are speaking about the tissues with special properties and embryonic connective tissue. And the next lecture will finish the tissues of internal environment, and we'll speak about the skeletal connective tissues, cartilage and bones. But now we'll continue and speak about the tissues with special properties, which are subdivided into the three main types: reticular, adipose, and pigment connective tissues. Reticular connective tissues, look at this, situates in the organs of hemopoiesis, where formation of blood-formed elements take place, and forms the specific environment for the developing formed elements. So we can see, for example, the developing lymphocytes, and around the developing lymphocytes, we can see the components of reticular connective tissue. Reticular connective tissue consists of reticular cells named reticulocytes and also reticular fibers. We talked about the reticular fibers 10 minutes ago. Reticular cells, reticulocytes, are the cells with processes. Look at this, cells with processes with a large light nucleus. The processes of reticulocytes connect with each other, forming the structure like a net. Reticular fibers are slim and branched, made by the third type of collagen, and reticular fibers are embedded into the cytoplasm of reticular cells. If we look at your slide, this is a lymph node. Lymph node is the organ of lymphopoiesis. In this organ, the lymphocytes finish their differentiation, and the stroma of the lymph node is made by the reticular connective tissue. In this case, we use hematoxylin and eosin staining. That is why we cannot indicate the reticular fibers. With the big magnification of the microscope, we can see only the reticular cells. Fibers are not visible. Reticular cells are characterized by the processes and by the oxyphilic cytoplasm. Processes of these cells connect with each other to form the loops, and inside these loops you can see the developing lymphocytes. This dark purple nuclei. Students, are you here?
Yes, perfect.
Everything is okay? Don't sleep, please. So we
Yes, Professor.
We can see the reticulocytes only with pink cytoplasm and the processes, but reticular fibers are not visible. I repeat that reticular fibers require the specific method of staining. If we use the salts of silver, impregnation or staining by the silver salts, we can indicate the reticular fibers. These black strips are the reticular fibers. In this case, we use specific method of staining, impregnation by the silver salts. So reticular connective tissue is one kind of tissues with special properties. Another type of tissue with special properties is adipose tissue, which consists of adipose cells, adipocytes, or fat cells and other cells. So the precursors of adipocytes is mesenchymal cells, which transform into the lipoblast, and lipoblast then differentiate into the two main types of adipocytes: multilocular adipose cells with many small lipid droplets in the cytoplasm, or unilocular adipose cells, which is cytoplasm is filled by one large lipid droplet. According to the structure of adipocytes, we differ two main types of adipose tissue. If we see the unilocular adipocytes, this type of adipose tissue is called white adipose tissue. White adipose tissue is widespread tissue in our organism, situates over the skin, surrounds the inner organs, in omentum, situates in omentum, in other regions of our organism. The main function of white adipose tissue is a storage of lipid, depot of energy, depot of water, storage of vitamins, different, and performs the supporting role. This is brown adipose tissue. Brown adipose tissue is represented by the multilocular adipocytes. Multilocular adipocytes houses many lipid droplets, and mitochondria of these adipocytes houses many pigments, cytochromes. That is why we can see brown or dark red color of this type of tissue. These adipocytes are smaller than the white adipocytes, and the main function is thermoregulation, production of takes part in the regulation of body temperature. Thermogenesis, hot, production of hot. Thermoregulation. In your micropreparation in the finger skin, you also can find the white adipose tissue. We can see the unilocular adipocytes. And this part of our organism is named hypodermis. It's not part of the skin, situated below the dermis of the skin and made by connective tissue. Also, in this slide, you can see another types of connective tissue. For example, this is a small portion of loose connective tissue. I can recognize loose connective tissue because of the large number of cells. We can see large number of nuclei of different types of cells. This part of the slide shows the dense, irregular connective tissue. We can see less number of nuclei because of the presence of less cells, and many fibers. These are the collagen fibers. It's also layer of dense irregular connective tissue which separate the parts of white adipose tissue. And the last type of tissue with special properties is named pigment tissues. Pigment tissues is represented by a large number of pigment cells, adipocytes. Look at this. Pigment cells form the pigment connective tissue. For example, pigment connective tissue situates in the eye bulb. For the iris of eye bulb forms the choroid. It's a proper vascular tunic of eye bulb and houses many pigment cells, and this tissue has the black color. So we told about the tissue with special properties. And the last type of tissue, the embryonic tissues, mesenchyme and mucous. These types of tissue we can see only in the organism of embryo. And I repeat that mesenchymal cells are the cells with processes which do not form the contacts, intercellular contacts together, and houses the many extracellular matrix. Mesenchymal cells transform into the another types of tissues and cells of the organs of embryo. And one more, mucous tissue. Mucous tissue, also embryo tissue, is characterized by the mucous cells and many large volume of